where we aim to make A-level biology as simple as a cell. So today we're understanding how cells become specialized through differential gene expression, producing active mRNA, leading to synthesis of proteins, which in turn control cell processes or determine cell structure in animals and plants, including the lac operon. So cell differentiation. So stem cells become specialized through differential gene expression. The genes are activated or inactivated depending on the type of cell it differentiates into. The mRNA is transcribed from active genes only, which is then translated to form the associated proteins. These proteins modify the cell as needed to make the cell specialized. And this process of cell differentiation, it is irreversible. So we've looked at transcription before, but eukaryotes use transcription factors to control gene expression. So a transcription factor is a protein that controls the transcription of genes, and it does this by binding to a specific region of the DNA. So what are activators and repressors? So transcription factors can either be activators or deactivators. Activators increase the rate of transcription, so they help bind the RNA polymerase to the DNA at the start of a gene. Repressors, on the other hand, decrease the rate of transcription. So you can tell the roles from their names. If that's one way to remember. So they prevent the RNA polymerase from binding to the DNA at the start of a gene, and this prevents the transcription of that gene. Sometimes the transcription factor binds to a section of a gene called the promoter region. And they can play the role of either the activators or the repressor. So that's another thing to consider as well. So what are operons? So these come up as past paper questions pretty frequently, and it's also that one topic that no one really understands. So yeah, make sure you guys understand this if you want to stay ahead of the curve. So in prokaryotes, the transcription factors bind to a section of DNA called operons. Also remember, it's for prokaryotes. This doesn't happen in eukaryotes. So that's another mistake a lot of people make. So operons are a cluster of structural genes that are transcribed together. And they can have a promoter region and an operator region. So some operons have regulatory genes that code for activators or repressors. So the lac operon is a very popular example of operon, and once again comes up in past papers so many times. And this is found in some bacteria. Again, remember, this is not in humans, this is in bacteria. And this is involved in the production of an enzyme called lactase and two other structural proteins. The role of lactase is to break down the substrate lactose as it can be used as an energy source in the bacterial cell. And this enzyme is only synthesized when lactose is present, hence energy and materials are not wasted. And an enzyme that is only synthesized when whatever it needs to break down is present, such enzymes are called an inducible enzyme. So that's just another fact you guys can know, or if you're interested. I mentioned the operons having both a promoter region and an operator region. So this is an example where you can see it in real life. Well, you can't see bacteria in real life, but this is just one example you can think of. So the lac operon consists of the promoter for structural genes, the operator. You have the structural gene LAC-Z, which codes for lactase. You have the structural gene LAC-Y, which codes for permease, which allows lactose into the cell. You have the structural gene LAC-A, and this codes for transacetylase, which assists in cellular detoxification by preventing re-entry of the non-metabolizable lactose. You won't see these definitions in your textbook, most definitely know from what I remember, but I just added it so you guys know what's going on. It's to add more context to what's actually happening. So the lac operon also has the promoter and regulatory gene LAC-I. and this codes for the lac repressor protein. The lac repressor protein can bind to the operator in the lac operon and the lactose. The role of the lac repressor. So if the lac repressor binds to the operator, the transcription of the structural genes are prevented as the RNA polymerase cannot attach to the promoter because the lac repressor is in the way. And if it's bound to the lactose, so the shape of the repressor protein distorts and can no longer bind to the operator. So what happens in the absence of lactose? So we briefly went through this a couple minutes ago. So in the absence of lactose, the regulatory gene is transcribed and translated to produce a lac repressor protein. The lac repressor protein binds to the operator region upstream of LAC-Z. And as a result of this binding, the protein RNA polymerase is unable to bind to the promoter region, and transcription does not take place, hence no lactase enzyme is synthesized. If lactose is present, so if lactose is present in the medium, the bacterium absorbs this lactose. The lactose binds to the second binding on the repressor protein, becoming denatured so that the repressor protein and the operator can no longer bind. The RNA polymerase now binds to the promoter region instead, and the transcription takes place as normal. And the enzyme lactase is produced, which breaks down lactose, and this produces energy that can be used by the bacteria. Professor Snap today. Hope you found this video useful. This is definitely one of those topics it's quite hard to get your head around, but it should make sense. So feel free to watch the video again to wrap your head around this topic. And one thing I would suggest is to like draw out a flow chart so that you can clearly see what happens. So for example, have lac operon as the heading of the flow chart and then split them into if lactase is present, these are the steps that happens. Sorry, if lactose is present, these are the steps that happens, and if lactose is not present, these are the steps that happens. So by doing that, you'll be able to visualize what's happening in the lac operon, and you're more likely to remember things. So yeah, that is all. And if you have any questions, let me know in the comments below and I will get back to them. And don't forget to like, share, and subscribe.